In this topic, we're going to discuss biosensors. So we're going to look at what are biosensors, what are the features of a biosensor, what are the uses. We're going to discuss glucose biosensors and dipsticks. So what are biosensors? These are analytical devices used to measure or detect a particular signal. They convert a biological response into an electrical signal. Types of biological molecules include antibodies and enzymes. These are immobilized in some way, for example, on a dipstick. So here you can see how an antibody has been immobilized, and as the antigen binds, a physical or chemical change is going to be converted into an electrical signal that is sent to the detector. Here's another diagram to show you how a biosensor works. The catalyst converts the substrate into the product. A transducer senses the physical changes and converts it to an electrical signal. The signal is amplified, processed, and then it can be viewed on a display screen. So biosensors should be highly specific, stable, cheap, portable, and easy to use by semi-skilled operators. So what are some uses? Well, they're used to detect pollutants. They're used in food production and ripeness. For example, they detect ethylene. They're used in herbicide or pesticide detection of the levels, blood glucose levels in diabetics. They're also used to estimate the concentrations of compounds such as urea, ketones, and cholesterol in blood and urine. Okay, let's have a look at glucose biosensors. These detect blood glucose levels in diabetics. They use the immobilized enzyme glucose oxidase. This enzyme is very specific, so glucose levels can be measured in the presence of other sugars. Glucose oxidase catalyzes the conversion of glucose to hydrogen peroxide and gluconic acid. An electrode can detect a drop in the oxygen concentration, which initiates an electrical signal proportional to the glucose concentration. Another way to detect the glucose level is to use peroxidase to catalyze hydrogen peroxide. The peroxidase enzymes are mobilized. The hydrogen peroxide will then oxidize another chemical and induce an electrical signal. Okay, let's have a look at dipsticks. These are strips of card or plastic with immobilized enzymes on the surface. An example would be to test for glucose. So the glucose oxidase, which is the enzyme, peroxidase, which breaks down hydrogen peroxide, and the colorless hydrogen donor are all immobilized on a cellulose fiber pad. A chemical that changes color is potassium iodide chromogen, which will go from green to brown. The dipstick not only changes color, but also gives a quantitative measurement of the level of glucose. So the more glucose that is present, the darker the color will be. The color can then be quantified using a standard color chart. And that concludes our lesson, the end.